Okay, so let's get started. So hello everyone, welcome to our today's Immune seminar. It's absolutely my pleasure to welcome our today's speaker, Dr. Susan Lynch from UCSF. Susan currently is full professor of medicine, as I said, in UCSF. Very brief introduction about Susan. So Susan received her undergraduate and graduate degree in microbiology from University College Dublin in Ireland, and and then before completing her postdoc training as a Dean's Fellow in the Department of Microbiology and Immunology at Stanford. So Susan actually joined the Department of Medicine, UCSF, as a faculty member, started her independent research program in 2007, and then all the way up to 2017, become full professor in Department of Medicine, UCSF. Susan's research program investigated the role of the human microbiome in chronic inflammatory diseases of GI and the respiratory tracts, with a focus on childhood atopy and asthma using ecological models. Her lab integrated high-resolution microbiome and immune databases from large human cohort to map microbiome immune dynamics over time and space and relate them to disease outcomes. Her key contributions, including linking infant gut and airway microbiota composition and then function to childhood asthma risk. Her lab has identified airway microbiome states predictive of allergy and asthma, and uncovered gut microbiome genes that drive immune dysfunction. So recent work from her lab shows how fecal microbiome and um, uh, metabolic profiles predict responses to peanut oral immunotherapy. These insights support development of microbiome-based interventions, including a microbiome drug from her lab, which currently in a phase 2 clinical trial to prevent childhood allergy and asthma. So Susan has served on a National Academy of Science and Engineering Committee, and as a National American Society of Microbiology distinguished lecturer. So she has received numerous awards, which including the Rebecca Buckley Lectureship and then Davy Perman Lectureship from AAI, and then was named as one of Foreign Policy Magazine's Global Thinkers. Her lab has published more than 200 papers, and in particular in 2016, Susan co-founder Cyota Therapeutic company as a biotech company. Their lead microbiome live biotherapeutic for the prevention of childhood atopy and asthma is currently in phase 2 human clinical trial. I think today Susan going to talk to us about her recent work about microbiome predict and prevent and treatment in disease. Welcome, Susan. Thank you so much to do this for us. We are very much looking forward to your talk.
Great. Thank you so much, Tran. And I'm really thrilled to join you today. As I was saying at the outset when we started our conversation, you guys are pretty hardcore that you're still online and want to engage in some scientific, at least listening to begin with, and discourse then at the end of the lecture. So as mentioned, my group is really particularly interested in asthma and in particularly in allergic asthma. The majority of children in the United States that have asthma have underlying atopy or allergic disease. And as you can see from this map of the kind of global prevalence of asthma across the globe, it really is not equally distributed. There are, industrialized nations where we really see the greatest burden of disease, suggesting that this is really a disease of westernized lifestyles and environmental exposures. Um, and while we can treat asthma, we have no cure. And so for many years, I have been really interested in understanding the genesis of the disease, the developmental origins of allergy and asthma in an effort to really move towards primary prevention. And as we know, the human microbiome evolves over our lifespans. In particular, in the first 3 years or so, in the gut, and we've also more recently shown this in the airways. The microbiome evolves. This starts from a very simple kind of community of microbes that are present. And this evolves over time. In the gut, we develop more diverse microbial communities over the first 3 years of life. And this, of course, occurs in lockstep with immune development and functional programming of immunity. What we know from epidemiological studies is there are a large number of early life exposures, either during pregnancy or during the first years of postnatal life, that increase or decrease risk of developing allergies and asthma in later childhood. And they include everything from cesarean section to formula feeding to antimicrobial exposure, maternal stress, exposure to livestock and pets in the home that influence the risk of disease development. And when we looked at this list, I became very excited because we know that many of these exposures shape gut microbiomes. Things like formula feeding, antimicrobials, obviously, cesarean section even influence the numbers and types of microbes that are present in the gut microbiome in early life. And the gut is where we house the largest numbers of organisms. In the distal gut, there are trillions of microbial cells. And their interaction with immunity has become very clear that they are shaping and guiding and influencing immune function in the gastrointestinal tract. And so, what this suggested to us is in order to understand a disease that's commonly diagnosed in childhood, most children are diagnosed with asthma in kind of, 3 years plus, a few very severe children earlier than that. But to understand this disease, allergies and asthma, perhaps we should be looking at a much earlier phase of development and actually interrogating the very early life gut microbiome that is educating immunity in the system as a means to understand who is on the trajectory to allergy and asthma development. And this was a little outside the box when we started this study to examine the gut microbiome to understand development of an airway disease. But we began by examining a birth cohort called the Queals birth cohort in the greater Detroit metropolitan area. We simply had 130 samples, stool samples collected from 1 month old babies. And this was a racially and socioeconomically diverse population, which is a really great way to study human populations, so your results are more generalizable. In this birth cohort, we knew whether these children went on to develop atopic disease at 2 years of age, and more specifically, asthma by 4 years of age. And we first began by just simply asking which bacteria were there using 16S ribosomal RNA sequencing. We also looked at fungi using another biomarker gene that we sequenced called the interspacer 2 region. But I'm just going to show you the bacterial data that we had. And rather going in with a kind of ordained idea of what we might see, we simply asked an algorithm called a Dirichlet multinomial mixture model whether we could see distinct 1 month old gut microbiota states that were reproducible across members of this population. And what we found was there were three distinct gut microbiota states, which we called neonatal gut microbiome 1, 2, and 3. And here I'm showing you what we call a principal components analysis. This really takes this massively multidimensional data set and reduces each one of these bacterial profiles that we have down to a single spot on this plot. And how you read this is the distance from any one of these spots to another spot tells you how similar the profile of bacteria is in any number of the samples that we have profiled. So what you can see is that these green NGM2 samples are distinct from the NGM3 samples and even these NGM1 samples. So what this tells us is that there are three groups of microbiologically distinct babies in this 1 month old kind of sample set that we have. And so it suggested to us that by 1 month of age, there are three groups of babies that have very different gut microbiomes. But the question was whether those starting life with those different gut microbiomes related to different clinical outcomes in this population. And the answer was a resounding yes. The children who had this distinct NGM3 gut microbiota composition which was about 11% of the population we studied, exactly the proportion we would expect to find in the US population that are at risk of developing allergy and asthma. That group was at significantly higher risk of developing atopy by age 2 or physician diagnosed asthma by 4 years of age. So we had now a 1 month old kind of predictor, microbiological predictor or correlate at the very least of disease development, increased risk of disease development years later in childhood. So what was different about this NGM3 gut microbiota? Well, it was depleted of a whole range of bacteria, really, and we had fungal enrichment, things like Candida and Rhodotorula, which we know in mouse models can induce allergic inflammation. Because of so many bacteria were missing from this gut microbiota, it really was functionally deficient. When we predicted the functions of these 3 gut microbiota states, the NGM3 gut microbiota was really deficient, predominantly in microbial metabolic pathways. So that led us to ask, are these distinct gut microbiota metabolic machines that are in these 1 month old babies? So we performed on targeted metabolomics and then did some comparative analysis across these 3 groups. And what we found was that this high risk for allergy and asthma group of NGM3 babies, their fecal microbiomes were depleted of a whole range of anti-inflammatory metabolites, including polyunsaturated fatty acids or PUFAs, steroid metabolites. And it was really a saccharolytic or a kind of a simple sugar loving microbiome. We saw enrichment of quite a number of simple sugars, which we now know can drive inflammatory phenotypes from immune cells. And they were highly enriched for this one oxylipin called 1213 dihome. Consistently, it was one of no matter what way we carved out the data, this lipid kept coming up as a really highly enriched lipid in this high risk group. But, these are all correlations. So we wanted to understand whether the products of the feces of these babies could actually promote features of allergic inflammation in vitro. All we had from these infants was stool. And so we generated an assay in which we took fecal samples, we generate a slurry with saline, and then we filter, sterilize that sample to take out all of the microbial cells. We're now asking whether the metabolic products of these fecal microbiomes can actually drive allergic inflammation in a model system in which we had basically taken healthy adult human PBMCs and purified the dendritic and naive T cells from those PBMCs. We co-incubate the dendritic cells with the fecal extracts, and then we wash them off, and then co-incubate them with the naive T cells and ask what they become when they grow up. And obviously, we have a particular interest in Th2 cells and T regulatory cells because that's the canonical kind of immune dysfunction that is characteristic of allergic inflammation. Increased Th2s, reduced Tregs, increased immunoglobulin E, and more B cells producing that immunoglobulin E. And so when we did this experiment, what we could find to our absolute delight was the fecal products from these 1 month old NGM3 microbiologically distinct babies could induce Th2 cells, promote increased IL-4 production, and reduce regulatory T cells. So essentially, we could recapitulate with these fecal extracts in vitro kind of characteristic features of allergic inflammation with these previously healthy dendritic and T cell and naive T cells from healthy adult donors. We next asked whether that lipid that I mentioned, 1213 dihome, which was highly enriched in the babies who were at the higher risk of developing disease, could recapitulate any of the features of the fecal extracts in this dendritic T cell assay. And what we found was that this molecule, in a dose-dependent manner, reduced the frequency of regulatory T cells and their capacity to produce IL-10. So the higher the concentration of this lipid, the fewer Tregs and IL-10 and anti-inflammatory capacity that the system has. That's all great, but it's all in vitro. The next question is whether we could prove that this lipid actually drives allergic inflammation in the airways. And we used this in a mouse model. So in this model, we had 3 groups of mice, and they were treated, one group was treated intraperitoneally with 1213 dihome right before each time we treated the airways of these mice with cockroach allergen challenge. This is a very traditional airway allergic inflammation model. So the question here is if you provide viperitoneal delivery of this lipid into the mice, if you provide them with this lipid, can you exacerbate airway allergic inflammation in response to cockroach antigen? And the answer was a resounding yes. Here are the control animals, no treatment with antigen and no treatment with 1213-dihome. Here are the animals in which we've induced cockroach allergen, allergic inflammation in the airways, and you can see in the bronchioles and around the blood vessels, we're seeing more kind of nucleated cells and inflammation. These are the animals that got the 1213-dihome before we hit their airways with cockroach antigen. And now what you can see is that we have basically obfuscation of the bronchioles, and there's this huge inflammatory cell influx into the airways. When we examined regulatory T cell populations in these airways, what we saw indeed is that the animals who received the 1213-dihome failed to mount the same degree of T regulatory cells in their airways, and we saw an uptick in their serum IgE levels. So this proved to us that this molecule delivered peritoneally to these animals during this kind of cockroach antigen exposure and allergic inflammation induction in the airways really could recapitulate the features of allergic inflammation that we'd seen in vitro in this animal model. So that got us pretty excited about where this molecule was coming from. And it turns out that 1213-dihome is a product of linoleic acid metabolism. Linoleic acid is an omega-6 polyunsaturated fatty acid that has become highly prevalent in westernized diets over the last several generations, right in lockstep with the allergic kind of prevalence that we have across these populations. And so typically it can be converted catalytically to 1213-epome by a cytochrome P450, or under oxidizing conditions, this reaction can occur spontaneously. And then this substrate 1213-epome is converted to 1213-dihome by a catalytic domain and epoxide hydrolase gene. So of course we hypothesized that it's the microbes in these gut microbiomes that are driving the production of this 1213-dihome molecule in the gut of these babies who are at high risk of developing disease. And so to search for these genes in the fecal microbiomes of these babies, we performed shotgun metagenomics in a kind of a case control study where we had babies who were at the highest risk and the lowest risk of disease development in our wheels cohort. And we used a tool called Shortbread. This is a tool that allows us to basically go in and fish for specific genes and quantify them. And we specifically went looking for epoxide hydrolase genes. And what we found was that in these babies, those who went on to develop atopy or asthmatic diagnosis in childhood had significantly higher copy numbers of bacterial epoxide hydrolase genes in their fecal microbiomes compared to their healthy counterparts. And that was coincident with the higher concentration of this lipid 1213-dihome in the fecal samples of these babies. But having a gene doesn't mean that having an epoxide hydrolase doesn't necessarily mean that it is making the specific product that drives this kind of immune dysfunction that we'd observed. And so we took the top most prevalent epoxide hydrolase genes that we had found in these fecal metagenomes. We cloned them into a plasmid, transformed E. coli, and then did an assay to test, first of all, were these epoxide hydrolases? Was this bioinformatic tool actually accurately identifying epoxide hydrolases? And could they basically convert glycidol to glycerol? Next, we asked about the specificity of these epoxide hydrolases and whether they could convert 910-epome to 910-dihome, or the target. Could they convert, which ones could convert 1213-epome to 1213-dihome? And what we found was they had variable capacity to act as epoxide hydrolases. They could all convert glycidol to glycerol. 2 of them could produce 910-dihome, and 3 of these enzymes could produce the target molecule 1213-dihome. So what this suggests to us, as you can see, these are in Enterococcus faecalis and Bifidobacterium bifidum. So this may be basically strains that have this gene, have the capacity to make this molecule, which is reducing Tregs, at least in vitro and in vivo in our animal models. And we think it's doing the same in babies who go on to develop disease development. The last part of evidence we had here was we took that E. coli that could produce the 12-13 dihome, the most potent 12-13 dihome producing epoxide hydrolase. And now we performed another mouse experiment. Here, instead of peritoneal injection of the lipid, now we orally treated mice with this E. coli. We supplemented them orally with this E. coli that could produce 12-13 dihome. And we give them linoleic acid because that's obviously the upstream substrate that we need to make 12-13 dihome. And then two of these groups of mice got cockroach allergen challenge as we'd done before. And what we could find was only in the mice where we provided them with E. coli that could make 12-13 dihome, did we see this increased 12-13 dihome production in the serum, produced in the gut, but found it in the serum of these animals, indicating that this lipid is not restricted to the gut, but gets translocated into the circulation. And therefore, this is the mechanism we think by which it's modulating immune function in the airways of these animals. And what we also showed was, again, as we'd seen with the peritoneal delivery of this lipid, if you provide animals with an E. coli strain that can make this lipid and you treat their airways with cockroach antigen and induce airway allergic inflammation, the group of animals that have the gut microbial capacity to make 12-13 dihome is the group that has this reduced regulatory T cell populations in the airways of these mice. So this was really for us the first example of gut microbial product that can be made in the gut, enter the circulation, and influence the regulatory T cell population in the airways upon allergic inflammation induction in these animals. So that's really wonderful. That shows us that T cell populations are impacted by this lipid. But we were also curious as to whether this lipid actually impacted macrophages, the other kind of antigen presenting cells. We had shown in that previous study that 12-13 dihome influenced lipid uptake, metabolism, and CD1A expression on the cell surface of cells, on DC cells. And we wondered whether it was just exclusively impacting dendritic cells or whether it could impact the behavior of macrophages. And macrophages are interesting because they sit at the crossroads of antimicrobial responses and antigen presentation. And we thought they were an interesting population to examine in the context of exposure to this lipid. So the first, very simple analysis we did is we just simply asked, if you expose macrophages to 12-13 dihome, does that shift their kind of functional phenotype towards an inflammatory phenotype and away from a wound healing phenotype? And that was our hypothesis, that this is a microbial derived product that is suppressing the capacity to downregulate inflammation, Th2 inflammation, and is also driving inflammatory responses that the microbes that make this are immune to, essentially, but would take out and prevent other organisms from colonizing the niche. And so we used, with flow cytometry, just IL-1 beta and CD206 positive cells, looked at their ratio to ask, basically inflammatory versus, maybe wound healing, max, what do we see? And that we saw that in a dose and time-dependent manner, this 12-13 dihome promotes these inflammatory IL-1 beta expressing cells. And these are controls where we can see with interferon gamma and lipopolysaccharide, we can induce kind of these IL-1 beta producing cells. I will say, though, I won't show the data here, we validated this and we did a qPCR over time, over 48 hours of exposure to this lipid, and showed exactly as we'd seen here, IL-1 beta come up, but then several other markers of inflammatory macrophages upon exposure over time. They came up around 8 hours post exposure, expression of these genes. We next asked whether, microbes are wily. They might want to promote inflammation that they are not susceptible to, but their competitors are. And they may also want to reduce the immune cell's capacity to clear these microbes these pathogens from the system. And so we looked at macrophage phagocytic capacity in the context of 1213 dihome exposure. And here you can see, irrespective of the cell line that we used, we see a significant reduction in phagocytic capacity of these macrophages once we expose them to 1213 dihome. And we'd shown this with a completely different assay as well, another way. This is really highly consistent. Lastly, we asked whether the inflammatory macrophage phenotype that we see induced by 1213 dihome is exacerbated by exposure to an antigen. And here we tried quite a large number of antigenic exposures: cockroach antigen, lipopolysaccharide from microbes, ovalbumin, and peanut. And we could consistently see that in every case, 1213 dihome induces these inflammatory macrophages. But once you then hit them with an antigen, you really significantly increase the number, the proportion of inflammatory macrophages that we see in these assays, suggesting that in babies that have these the capacity, their gut microbiome has the capacity to produce this lipid, that once they start seeing food antigens and even microbial antigenic stimulation, that drives greater inflammation, which we think drives damage to the system and really shapes the kind of microbiome development and immune priming that's occurring in the gut in these babies in very early life. We next asked whether kind of changing macrophage phenotype has a downstream knock-on effect on T and B cell phenotype. So again, this is a co-culture assay. Instead of using dendritic cells, we're now using MAC as the antigen-presenting cell type, co-cultured with naive T cells and B cells from, again, all purified from healthy adult donors. And here we're now exposing these to with or without 1213 dihome in the context of peanut antigen stimulation. And what we found when we bring this assay out, it's almost like a month-long assay. What we found was that the memory B cell to naive B cell ratio is significantly increased when we expose the max to 1213 dihome in the context of peanut antigen exposure. And also we see this shift towards greater IgE production, less IgG production in these B cells in this assay. So what this suggests to us is that, modulation of macrophage function can have a knock-on effect on the B cell, memory B cell population and their capacity to produce IgE, particularly in the context of peanut antigen exposure. So the next question we had was, how is this early life exposure having such a long-lasting effect on immune function in these infants? And we hypothesized that perhaps it's due to epigenetic modifications of these really key immune cell populations, the antigen-presenting cells. And so in this study, we exposed macrophages again to 1213 dihome, our vehicle. What was really striking to us is there was a really consistent number of regions of the macrophage genome that were epigenetically modified. This was a TAC-seq that we performed, though we also did methylation analysis, and both showed very similar results. But here I'm showing you the TAC-seq data, suggesting that this lipid really has a very strong kind of target, epigenetic targets, gene targets for epigenetic modification in the genome of macrophages. And when we did transcription factor binding site analysis of these regions of the chromosome that are modified epigenetically, we were really struck by they were all interferon gamma response elements. And so we set up another experiment to ask whether there's a functional impact, a transcriptional impact of exposing macrophages to 1213 dihome, specifically on interferon regulated genes. And we did this experiment where we took the max, exposed them to plus or minus 1213 dihome in the context of peanut or lipopolysaccharide exposure. These are the interferon regulated genes that we just did qPCR on these. Just is there a transcriptional impact of these epigenetic modifications at these sites? And what we found with lipopolysaccharide is upon exposure to 1213 dihome and lipopolysaccharide, we saw a number of these interferon regulated genes and regulatory factors, including USP18, viperin, interferon, IRF1, IRF7, were downregulated following exposure of the macrophages to 1213 dihome, suggesting that the MACs have a reduced capacity to respond through the interferon axis to microbial lipopolysaccharide. We saw the opposite with peanut antigen exposure. Here we saw a very different group of interferon regulated genes or regulatory factors, and they are all upregulated in the context of peanut antigen exposure. So it suggests to us that this lipid is muting macrophage, interferon regulated macrophages response to lipopolysaccharide and enhancing inflammatory interferon regulated responses to peanut antigen exposure, really the perfect storm for allergic disease development. And this was work carried out by Lynch. It was published last year in Journal of Immunology Research. So this is all great. We're starting to get to grips as to how this molecule may be shaping not just one or two cell types, but really seems to be influencing antigen-presenting cells in particular to shape immune phenotypes. That's all great, but it's in vitro and it's in model systems. Does this molecule really matter for disease development in children? And so to really validate what we'd seen originally in a very small cohort, we took two independent cohorts and we quantified the number of epoxide hydrolase genes in the fecal microbiome of those children and the quantity of 1213 dihome in those fecal samples. And what we found in these two independent cohorts is that for every doubling of the copy number of bacterial epoxide hydrolase genes, the odds ratio of developing either atopy or eczema or asthma in these children significantly increased. And essentially what we, just to put this into real numbers, babies that just have 4,100 copies of these bacterial genes have an odds ratio of 15 times more likely at 1 month of age to develop asthma and atopy phenotypes much later in childhood. But remember, having a gene doesn't mean it's expressed. So instead, we looked also at the lipid itself. And here we found that again, for every nanogram increase of this lipid in the feces of these children, we saw a significant increase in risk of developing disease in much later childhood. Here it was even more compelling. Babies that had just 2,000 nanograms of this lipid in their feces, 55 times more likely to develop disease in childhood. And I can tell you this was not the highest concentrations that we found in these babies. So we think collectively, this tells us that we really have a molecule here that profoundly influences immune function. And I think the other data that we have to suggest this are two very new studies. One, it just got accepted in Nature Communications last week. This is a trial of children on peanut oral immunotherapy. And there we see that amongst the metabolites that are most highly correlated with peanut IgE is 1213 dihome in that population. In a completely independent cohort, and this is work we're preparing for publication, we see that babies who are most susceptible to infection, microbial infection in early life, are significantly enriched in their fecal microbiomes at 1 month of age, and 1213 dihome is our top hit in that cohort as well. So consistently, irrespective of the cohort, the relationship with allergy, in particular with peanut allergy, and the relationship with increased susceptibility to infection holds across our cohort. So we're pretty excited about this. I think that this is just one example of a fecal microbial derived metabolite that promotes immune dysfunction and that is consistently for us across cohorts associated with childhood allergy and asthma development. And in fact, I spoke to somebody a couple of weeks ago who has a 4,000 strong cohort, and they are also finding 1213 dihome associated with asthma in that cohort. So we're pretty excited because we're starting to understand why this might be a really critical molecule and how it modulates immune function at a cellular and at a molecular and an epigenetic level. But I want to just talk a little bit about, what's happening across time. This is kind of the temporal aspect of how we think about disease development and microbiome development. And this is data that we generated some years ago on a cohort, birth cohort again, where we had meconium samples and stool samples collected from babies at birth 1, 3, 6, 9, and 12 months of age. And here I'm showing you another one of those principal components analysis plots where each spot is a bacterial profile from an individual baby, and the distance from one spot to the other tells you how similar the microbial profiles are. And here you can see that there is a significant difference in the meconium microbiota, which forms in utero of those babies that are healthy, are very low risk of developing disease, and those that are high risk. And they're at high risk because they have at least one parent that has allergies or asthma. And what you can see is they start life with a different group of microbes that, again, we think is priming immunity, and they follow a different trajectory of microbial development. They are delayed in their microbial diversification, and we think it's because this is a pathogenic microbiome that is driving inflammation that it is not susceptible to, that keeps the other commensal organisms from colonizing the niche. And I will say what's really gratifying is this work was published in 2018 and it has been validated in much larger cohorts across the globe, in Canada in the child cohort, in COPSAC in a European cohort. So this is now a characteristic feature of the trajectory to allergic disease development, is showing this delayed maturation and diversification of the gut microbiome in early infancy. And so when we saw that, that made us consider, well, maybe 1 month of age is not early enough. Maybe we should really be dialing back this timeline into pregnancy. And that's because several mouse studies have demonstrated that influencing the gut
你
microbiome
好，
during,
拿到实际了吗？
oh, I'm sorry, is there
拿
a
到实际图
question?
触发成以后门出来啊。
I'm sorry. Thank you. So there are studies from mice showing that manipulating the gut microbiome during the gestation period alters immune function in the offspring. So we really want to get at this from a microbiological and immune aspect in humans. And so to do this, we examined a cohort of 172 paired maternal vaginal samples and infant stool samples. And the stool samples were collected at 1 to 2 months of age in these babies. We had quite a number of maternal factors that were measured: asthma, eczema, mode of delivery, etc. And our outcome in the babies is whether we could detect IgEs in their serum at 1 year of age, if you can detect that they're really on the trajectory to allergic disease development. We performed 16S sequencing on all of these samples to find out which bacteria were there. And for a subset, a small subset of them, we did shotgun metagenomics to understand what genes were encoded by these microbiomes and how they related to outcome. And so what we found was that the vaginal microbiota in the prenatal period at 36 weeks gestation forms 4 different vaginal clusters. Again, this is the same approach that the data tell us what's there instead of going in with preordained ideas. And in fact, this has been shown in multiple other, even larger studies. So we could see 4 vaginal microbiota, distinct vaginal microbiota states. And what we found was that these related to environmental exposures of the mother. Everything from whether they lived on a farm, which, remember, relates to protection against allergic disease development, or to what they were consuming, their diet, and whether they had depression or stress related to their vaginal microbiota composition. So suggesting that these environmental, dietary, health status factors can shape the types of microbes that are prevalent and present in the vaginal tract right before delivery of the baby. And of course, delivery of the baby is what dictates which microbes are transmitted from mother to child. And so here I'm showing you a heat map of the shared amplicon sequence variants, or 16S sequences that are shared between mother-infant dyads. So each column is a mother-infant pair. And if there's a red rectangle, it means that that organism was found both in the mother's vaginal sample and in their paired infant sample. And you can see some of these things like Bifidobacterium, Staphylococcus are they're more frequently shared between mother and child. But the next question we had is whether the variance in this pattern of shared microbes across this population related to the mother's health status and to whether she had allergies and asthma and to whether the baby developed early markers of allergic disease and IgE, elevated IgE in their serum. So we related this variance in this pattern of sharing and found that it significantly related to the vaginal microbiota cluster. So the type of vaginal microbiota that's present prior to delivery dictates the types of microbes that are shared and are present and detected still at 1 to 2 months of age in the baby's fecal sample. Phenomenally for us, we also found that the mother's prenatal health status, whether she has diagnosed eczema and asthma, and her pre-pregnancy BMI related to the types of microbes that were transmitted to the baby. And the types of microbes that were transmitted related to the birth weight of the baby, their mode of delivery, which makes perfect sense, and to their total IgE in the circulation, so their early marker of allergic inflammation. And so we weren't happy with just showing that this was the first real demonstration that there might be a microbial inherited component of allergic disease development. We wanted to understand functionally what might these babies be inheriting that may be protecting them against allergic disease or contributing to allergic disease. So this is data from our shotgun metagenomic analysis. Here, what I'm showing you is a machine learning analysis of the shared functional features between those pairs of samples that we analyzed. And what you can see is that in the babies that do not have a detectable IgE, so they're less likely to be allergic, they inherit microbial capacity to metabolize stress hormones. Remember, stress was one of those things that was associated with the maternal vaginal microbiota. They also can degrade tryptophan, which is one of the most potent immunomodulatory molecules that signals through the aryl hydrocarbon receptor. And they inherit their own microbial antihistamine. So, if you've got allergies, you take Claritin, you take some antihistamine. Babies who don't develop elevated IgE concentrations in their circulation by 1 year of age have microbes that are capable of degrading the Th2 promoting histamine. On the flip side, the babies who do develop disease, they inherit microbes that produce fructose from glucose. That's that glucose 6-phosphate isomerase. And we know that fructose enhances inflammatory response to bacterial lipopolysaccharide. And they also have suggestions that they may be pathogenic microbiomes. We see pre-pillin peptidase, pili can drive inflammation, although we're expanding this very largely. We now have a cohort of almost 200 mother-infant dyads where we're looking far more broadly now at these traits. But this has us very excited. It suggests to us that it's not just human genetics that matter. And to be quite frank, human genetics have not explained the prevalence and the rapid increase in allergic disease and asthma development in westernized populations. But what this data suggests is that there is actually a microbial inherited component of allergy and asthma heritability. And that these data are starting to allow us to understand what may be protective against disease development and which microbial activities may be driving immune dysfunction that promotes allergic disease development in early life. And so this is kind of the model we have right now. I didn't show you all of the data for this, but what it suggests to us is that we really need to be thinking about the prenatal period and the maternal exposures rather during this period that may shape microbiomes in the gut, in the vaginal tract, and then how that relates to what's transmitted to the baby. Not just microbes, but immune features that are transmitted in breast milk, for example, immunoglobulins that are transmitted that will be related to the mother's microbiome and to epigenetic features that are transmitted. I don't have time to, there's a study we've shown where we've linked cord blood epigenetics to the maternal immune status and to the microbiome development of the airways of children over the first 3 years of life and their development of asthma in later childhood. So we think that much of this immune priming is occurring during the gestational period, and then that is in the microbes and the immune priming that's occurring and that is present at birth dictates the postnatal microbial immune development and functional state, and thus the trajectory to allergic disease and development. And so in the last 2 or 3 minutes, I want to go through what we're thinking about this, because I know, Tran, you mentioned this at the outset of some of the things we're doing. This is the work that we've been doing to ask, can we reinstate microbial functions at birth over the first year of life to re-engineer microbial metabolism in the gut microbiome, to shape immune development in that critical first year of development, to prevent disease development. And this is based on functional traits that we found to differentiate between babies who are at high risk or low risk of allergy development, identified the functional traits that were deficient in those babies, and then we designed a cocktail of microbes based on the smallest number of microbes that encoded the broadest breadth of those functional traits that are missing in those gut microbiomes in those babies. And this, as was mentioned, is part of a phase 2 trial that's wrapping up, where we're using this multi-species microbial biotherapeutic to treat high risk for allergy and asthma babies daily with an oral supplementation from birth. We're treating them daily for the first year of life, asking if we can mitigate allergic eczema as our primary outcome, but other allergic conditions as well, and asking if that's a sustainable effect. If we program immunity in early life, is that enough to kind of keep the ball rolling and have a self-sustaining microbiome immune crosstalk that prevents disease development? The other things we're looking at is trying to understand, how do we become more targeted in how we're thinking about disease in our population. This is the airway microbiome across a very large number of samples in 413 children with asthma. Again, we can see there's largely six very different airway microbiota states that we can see in those children, and they are significantly related to clinical outcomes in these children. In particular, our younger patients who are exacerbation prone, that we found very difficult to treat, and who have eosinophilia in their airways and markers of eosinophil activation, eotaxin, for example, they tend to be dominated by Moraxella. And so what we're now doing is developing CRISPR-Cas-based therapeutics to specifically eliminate the pathogenic Moraxella catarrhalis in the airways of these children to prevent them from having exacerbations, because the children who have Moraxella catarrhalis in their airways during a respiratory infection are the ones who go on to exacerbate. And hot off the press, it's not even on the press yet, but data we've generated in the airways where we've developed a protocol to enrich for the microbial transcriptome in these samples. What we've shown is that in children with asthma who have a respiratory infection and have Moraxella in their airways, the Moraxella is actually expressing leukotriene D4, one of the most potent bronchoconstrictors that there is during respiratory infections that progress to exacerbation. So we really are going after very specific targets here, and we think that we might actually change how we think about what's driving exacerbation in these children and develop therapeutics that are really tailored to the specific microbial activities that drive exacerbation. And then the last thing we're interested in is really thinking of, microbial drugs. How do we get at microbial products that modulate immune function for the better? And so this is a study that's ongoing where we've taken infant samples, we've done metabolomics on them and asked which metabolites actually are associated with protection against asthma development at 10 years of age. And these are 1-month-old infant stool samples, asking what protects asthma in childhood. Mass spectrometry, identify asthma protective metabolites, and then we screen them with a macrophage reporter cell line to ask which metabolites can actually reduce NF-kappa B inflammation in the context of, in this case, peanut antigen. We've done lipopolysaccharide as well. This is one of our lead compounds. You can see as we increase the concentration of the compound, we really see a significant decrease in interferon gamma, NF-kappa B expression by these MACs. We've taken this compound and we've asked whether it can quench basophil activation, and it does. And it looks like it's working through the IKAPA kinase pathway, which is super exciting for us because that brings in the whole oncology side of things. We've identified which bacterium makes this. We've isolated it. We've shown by mass spec that they can make this organism can make this molecule. And now we're going into preclinical studies in mice to ask if we induce allergic inflammation in these animals, can this molecule or the microbe that makes it mitigate basophil activation and allergic inflammation in these animal models. And this is just kind of one of our kind of pipelines to identify microbial metabolites that may actually modulate immune response and prevent disease development. So with that, I just want to acknowledge everybody who's on this slide. It takes a village to do this kind of work. I'm internally grateful for my collaborators who are just wonderful. And of course, to the NIH for being so supportive of the work we do. And with that, I thank you for your attention, and I'm happy to take any questions you may have.